I, I've called him a force of nature many times because he is. He's probably the most recognized clinical figure in the geroscience field and doing so much in so many different ways. You're just going to see one small example, or one of maybe several examples in this session. But Nir has been a great supporter of this conference. He flew over for the London conference back in 2017 and has appeared in every other conference since. And he's always generous with his time for other conferences. So it's a real honor to have Nir as a real collaborator in this effort. And he's a dear friend. Nir, I will only thank you, and I'll stand by to participate in this session, though I'm just going to be on the outskirts. And call me when needed.
Terrific. No, you can stay on. And you know, I told you flattery will get you nowhere. You're complimenting so much. I already agreed to do this session, you know.
I know.
I also want to tell you guys, I love targeting healthy longevity. And in fact, I suggested in 1917, I suggested health span and longevity as a title. And
Yeah.
the argument we had there right, it was near Wembley,
Wembley, yeah.
near the dinner table, is yeah, but what about obesity? And my point was obesity accelerates aging. Well, that's what it does biologically. So you're coming around,
We're slow learners,
but I'll tell you one thing. And I'll tell you who's we in a second, established 2 weeks ago the Healthy Longevity Medicine Society. We are going to start to train doctors that are interested in longevity, educate them, and be very conservative in how we do it. It's pushed by Andrea Myers, who I believe I introduced you last year, Evelyn Bishop, who's a professor in Shanghai and in Switzerland, and really closely watched by Jim Kirkland, Tom Rando, and myself. So there's the biology aspects and the MD aspects, so we can make programs. So I think the name of Healthy Longevity is around, and thank you very much.
Well,
I'm here as you see by the background, as the scientific director of the American Federation of Aging Research, AFAR, that, as many of you know, have started the geroscience field, supported the research, spent $200 million to form the pipeline that we're talking about, at least the pipeline of mechanism leading to even some drugs, and also supporting the next generation of scientists that came through. I started with a $35,000 grant with AFAR, and so are many of us, Tom Rando and Jim Kirkland including, and that's how we got to the time we got, and that's why we're so indebted to AFAR. And apropos creating the next generation, you guys, Thomas and Zen, are behaving like a next generation. I mean, the technology that you're using, your vision is very, very inspiring. And Joan as an addition. And I want to pick on one of the things that the panels in the morning said. We scientists are very bad at explaining things to the public. And so this is a trial here. We're showing no slides. I notice you ask people to have 5 slides, but in every slide they sometimes have 20 tables and figures with individual data, right? We have no slides. We have clear messages. We're all going to talk plainly. So this is what we're talking about. The American Federation of Aging Research, beside supporting all the pipeline, in the last few years, we started to do major initiatives that we think are needed in order to carry on the field. One of them is TAME, which we'll talk tomorrow, or maybe somebody will talk tomorrow. It's about basically demonstrating to the FDA that aging can be targeted and doing a lot of good things by doing that. The second thing is the FAST program, the FAST biomarker program. The FAST stands, I mean, you'll have to listen carefully now, for finding aging biomarkers by searching existing trial. There's FAST somewhere there, and we'll get to that a little later. And we'll start with the SuperAgers initiative. And let me tell you what's the SuperAging initiative, and then we'll jump into and introducing our panelist. So the idea is to recruit 10,000 centenarians and their families. This is an initiative that, by the way, is going to be very expensive, and it's supported by an individual who's 30 years old, and his name is James Fickle. And why do we want to recruit centenarians, and what do we want to do with them? We want to find all the longevity genes that we could do in order to develop drugs for them. What do I mean? Several pharmaceuticals, but mainly Regeneron, have discovered that a lot of what we were developing as drugs came from animals. Okay, we kind of thought that everything we find in mice is relevant, and we failed 95% of the time. And because of what we can do with genetics, pharmaceuticals said, let's find a genetic abnormality and develop drugs only if we know what does it do with humans. You have genetics, you have SNPs, you're right, you have changes in the genes that could have functional consequence of either a gain of function or loss of function. The best example is PCSK9, okay? And PCSK9 really was, it's in 2006 that loss of function in subject with a certain mutation was associated with 88% reduction in coronary heart disease. And in 2012, there was a drug, a phase 3 trial in New England Journal of Medicine. The distance between having genetics and develop drug and knowing that it will work is much faster when you have that. From an aging perspective, we found a functional mutation in the gene of ApoC3 that was in centenarians. Alan Schuldiner and the Amish found in 2008 also a mutation that was associated with longevity. In 2015, there was a drug developed. This is the most important sentence I'll say today. 67% of the drugs that were approved by the FDA in 2021 were based on human genetics, okay? That's why it's so important. So we want to use centenarians to find longevity genes, and that's why we're doing the initiative, and we're going to sequence the whole exome of 10,000 families of centenarians. So now I'm introducing, I'm the PI, I'm the guy who watched from afar, but now I'm introducing you the principal investigator of this, which is Sofiya Milman. And Sofiya is an endocrinologist. She's an associate professor of medicine and genetics at Einstein, and she has experience working with cohorts of exceptional longevity. So hi, Sofiya, and thank you for the short notice here. And I want to ask you first thing, before we go to the initiative, do SuperAgers have only longevity, or do they have health span also?
All right, thanks, Nir, and thanks everyone for the invitation. So as Nir is alluding to in his question, what we have found, and what many have found who have studied centenarians, is that they don't only live long, but they also live healthier, so that they get the diseases that we typically associate with old age, like diabetes and cancer and heart disease and Alzheimer's disease, 20 to 30 years later than people in the general population. And that's what makes them so interesting to study.
And I would add on that they also have contraction of morbidity. They spend very little time sick at the end of their lives. The CDC have followed since 1993. You can get the data yourself. Have showed that the last 2 years of medical cost of somebody who died over the age of 100 is third of those who die when they are 70. And that started to be the rationale for the longevity dividend. Of course, recently Andrew Scott comes and says, hey, you're way off, because it's not only that they're not in the hospital, what are they doing? They're traveling, they're shopping, they're buying things, so their value for their economy is even larger. So yeah, so they're healthier. Okay, good. So can you give an example from your studies of the effect of genetics, the effect of genetics on longevity? Start and I'll see where you stop.
Sure. So in general terms, what we've and others have been finding is that longevity tends to run in families, right? So the children of centenarians tend to live longer and also be healthier. And that suggests to us that genetics play a significant role. Of course, we can't rule out the possibility of environment, right, without looking at it, because they're also living in the same household, right, breathing the same air, eating the same food. But when we compare the lifestyle of the children of centenarians to the lifestyles of the children of non-centenarians, right, the general population, we found that there were really no differences in their diets, in their rates of smoking, in their exercise habits, alcohol consumption. Any lifestyle we looked at was really no different between the children of centenarians and the children of non-centenarians. And yet we found that the offspring of centenarians were protected from cardiovascular disease, from heart disease, compared to their peers. And so that was one of the examples that very clearly showed the role of genetics rather than environment that longevity plays.
And I would add that in centenarians, in our study, over 50% were obese or overweight or obese. 60% of men, 30% of women were smokers. Less than 50% did even moderate exercise, and they were very similar to the cohort of Enhance 1 study. So it wasn't about their it wasn't about a lot about their environment. Tell me, what are some of the genes that you're interested in, and what is your finding? Just to show, what I'm driving is to show that there's promise to what we're trying to expand, right?
Sure. So I guess some of the genes you've already mentioned, so we won't go over those. But I think what's really exciting is that some genes tend to show up in different organisms, across the evolutionary spectrum, right? There's certain genes that control the growth hormone pathway that have been shown to prolong lifespan, but also extend health span in organisms starting from yeast, to worms, to fruit flies, to mice, and it seems to play a role in humans as well, because there's several centenarians who've been found to have genetic variants that affect the growth hormone pathway and reduce the signaling through that pathway. And so what's really exciting, for example, about this pathway is that it seems to be consistent across nature. And so that gives us a lot of excitement to pursue that particular avenue for research.
Right, and we actually transferred it now back to mice and showed how we can target those mechanisms and have longevity in mice. Before we go to the study, just tell me, you're seeing people between 65 and, 112. Do men have menopause?
So interestingly, in a very recent study, we found that indeed they do, right? Which is not something that's well known. And one of the reasons we feel that it was not known is because they tend to develop menopause in their late 80s, right? So many men don't live that long. And when they do, probably no one looks for it. But we have this cohort of centenarians in whom we discovered that over 50% of men over age 90 actually have menopause, the classic menopause that we see in women with their testosterone dropping and their hypothalamic and pituitary hormones rising in response.
Do they have
So
hot
very
flush?
classic menopause symptoms. What is it?
Do they have hot flush?
We didn't ask because we didn't expect, right? We didn't expect to find it. So we never asked them, but we certainly will do that next time, right? When we do a more prospective study.
Okay, great. So Sofiya, what are the objective of the SuperAgers initiative?
So the SuperAgers initiative, which is supported by AFAR, really its goals are to increase the number of centenarians that we have for research, the genetics of centenarians for research. So this will be the largest cohort in the world. We're aiming to recruit 10,000 centenarians in addition to their offspring and control groups, as Nir has already said. And that will become a resource for the research community to use to study their genetics, their health history, primarily genetics, so that's what we're interested in, to identify genes for healthy longevity. And right now we don't have a cohort that's really large enough in order for us to be able to identify sufficient number of genes with enough statistical confidence.
And what are the challenges? I mean, John will tell a little bit about it, but what do you see as your main challenges as as it goes to the field?
So this is, I think, the first study of its kind, at least that I'm involved in, where we're going to be doing most of the data gathering using online tools. In order to expand the study to 10,000 individuals, we really need to be creative, I think, and utilize into the internet and technology. And so we'll be asking people to sign up using these technological tools, answer questions online. They'll actually be sent, they'll be mailed a saliva kit that they will send back to us, and we will use that saliva to isolate their DNA. So it's a kind of no contact study, right? Which I think we learned from the pandemic, that we have to learn to do things differently, especially when we're working with older populations who may be more vulnerable, and also because we're trying to really expand this to the largest population, I think, that has ever been recruited for a study like this. So we anticipate challenges with technology, right? So we're really going to be counting on their families to help them, their children, their grandchildren, maybe great-grandchildren. We'll have people standing by to help them as needed on the phone. But really looking forward to it. We think this will be a very exciting initiative that will be a wonderful resource for the entire research community.
Terrific. That's very exciting. I want to get John Beilenson into this discussion because really to talk a little bit about, look, when you have AFAR is American Federation of Aging Research. Every society have their own people that they target, right? You have the Diabetes Association, you have the Alzheimer, the Parkinson. How can we do something that is unique for aging? And John is kind of behind those issues. And I want to say, first of all, John has COVID. So he has singleness of purpose and he's really under fire now. And thank you for doing that. And I would say that all you have to know about John, what he's doing is you see it at his email and it says connect, communicate, and change. Right? John, if you want to say something else about what you're doing, say it now.
Well, thanks, Nir. And again, I appreciate that. Again, sometimes the limitations or the constraints of the sort of this online Zoom technology can work to one's advantage, so I don't have to put anybody else at risk. And again, I'm feeling reasonably well here and happy to join today. Yes, as Nir noted, I run a small communications and public relations firm, and our tagline is Connect, Communicate, Change. We work with a variety of nonprofit organizations, foundations, and government agencies to help them use communications to create social good, social change, social justice. And we have been working with the American Federation for Aging Research on and off for the last 30 years, have worked with Stephanie Letterman, the executive director, for many, many years, have known Nir for many, many years. And we're extremely pleased to be working on this project, which is not only, I would say 75 or 80% of our work is focused on aging and older and serving organizations that serve older adults. But this project in particular is really exciting because it proposes some unique communications challenges, recruitment challenges that we're looking forward to digging into and to overcome.
So Sofiya started describing the fact that you need to involve everybody in the family
Yes.
to do that. So it's a complicating challenge, but how would you describe your general approach to the superagers and their families?
Well, I think there are two things. One is exactly that we are, as opposed to recruiting for other types of studies, we are not directly recruiting the centenarians or superagers. And actually, to be accurate, our superagers, and I will say this is important for us in terms of the numbers, our superagers are people 95 and over, which actually adds to the end becomes actually considerably bigger in those 5 years, which helps the sort of the stomach ache that I have about trying to actually get 10,000 people in the study. But the first thing is that we are recruiting through intermediaries, right? So the family members, the children and grandchildren of the centenarians, and we're also going to be recruiting through folks at senior centers and at senior living facilities, et cetera, who know centenarians and oftentimes may be celebrating them in different ways. So it's an indirect approach. So that's the first thing, which again complicates things somewhat, but again I think is important and needed in this case. The second thing is that, in terms of our general approach, we're trying to sort of operationalize this question: do you know someone who's a superager? So I can't tell you. I mean, we've been planning for this project since March, or the beginning of this project since March, and this makes great cocktail party chatter. And people ask you what you're working on. I say, oh, I'm working on this really interesting project around superagers. And every time, I'm a person of a certain age, my cohort has parents, who are oftentimes in that age group. And so what we're trying to do is to figure out a way to ask the larger community, do you know someone who is a superager? Provide a community where the folks who are interested in and know the superagers can join, and then to connect them to this family study that will comprise the scientific basis of the work.
So really the first thing we're trying to do is we create a registry of basically superagers that will be really unique. We're not getting the sick people, right? It's we're getting the people who survived healthy.
Yeah. So I mean, I think there is some. I will say the study, as you know, is open to everyone. Assuming with the normal constraints around cognition and people's ability to fill out the forms, et cetera. But yes, for the most part, we are going to, and as already been noted, the rates of sort of healthy aging and health span are greater in this population than they are more generally. So, but that said, we are looking for those folks, and people are quite proud of the people we have recruited thus far are quite proud to recommend their parents and grandparents or their friends or the people who are in their facilities to be a part of this endeavor.
What are you doing as far as old age facilities? How do you get there? And B, what do you do with media?
So those are two of the big the big sort of vectors for us. We are looking at both the tops of senior living sort of chains to try to develop relationships that can get us sort of wholesale wholesale numbers of people who are in our age group, and we are looking and talking to individual activity directors and executive directors of senior living facilities to engage them and to get the 5s and the 10s and the 15s people that way. We're still, I will say, in all of this, we are experimenting, and in this early phase, we are trying to figure out what is the most efficient way to get people in, because to get to 10,000, we're going to have to replicate this process, and if it costs us, $1,000 to get each, a single person in a certain way, that's not a strategy that we can then likely use going forward. So we're looking for the most cost-effective ways to get people into the study.
Right. So that's a little bit highlight of the project and the challenges. But of course, if we'll have the genotype, it will be amazing. Let me give you one example of the importance of what we find with genotypes. So, there is a genotype for Alzheimer that's ApoE4. It wasn't mentioned actually today. But, the textbook will say if you have ApoE4, you're likely to be demented at 70 and dead at 80. Mm-hmm. So we have, so first of all, we have to know, please note that, you can be born with ApoE4 and you're not going to be demented when you're born or when you're 1 year or 10 year or 20 year, right? You need aging in order to get this disease. But those guys, I have 2 guys who are ApoE4, who are centenarians and ApoE4, and they're not demented. So the point is that those longevity, if you can slow aging, which is what those centenarians are doing, you can harbor lots of genotypes, and we have almost 5 genotypes for every centenarian that is associated with the disease, and they usually don't have that. So it's really important to find those genes that slows aging and allow them even to have other genes that can kill them, and they don't, because the longevity is so dominant. So, Sofiya, anything else that you think we've missed in this discussion that you want to talk, or last words?
No, I mean, I think there were many talks, right, over the past 2 days that I probably highlighted very well the fact that it's not inevitable, right, to age with disease. And I think centenarians or superagers, right, are a perfect model of that. They show us that it is absolutely biologically plausible for humans to live long and remain healthy, right? That it's not science fiction. There are people who are capable of doing that. And when you see that, right, then you I think you begin to believe it. And that's why I think identifying those biological pathways directly in this population, as you alluded to, Nir, is absolutely valuable. And so what we'll be doing, and there was a question to that effect, is we'll be collecting medical information about these individuals, family information, and we'll be getting saliva from them that will then be sequenced for DNA. So we'll get whole exome sequencing on the DNA of all these individuals, and then all the data will be posted on an NIH protected server. So the data will be protected. But any legitimate researcher who wants to use that data for research will be allowed to do so once they submit appropriate paperwork. So that will really democratize, I think, this data to almost anyone. And I think the more people are working on it, right, has been said before, the better, right? Because people have different ideas. And so the more researchers we can get involved, the more hands will be working on this data, the better for all of us.
And John, anything we've missed that you think is important to note? It's only the tip of the iceberg of whoever is involved in this project, but do you think that we should say something else?
Well, just one of the pure joys of working on this project is that I've been actually charged in part with telling some of the stories of some of the superagers. So we've had the opportunity to talk to a couple of folks. We just talked to a woman named Hilda. I won't say her last name. Maybe I just should protect her identity. But who is 100 years old and lives a tremendous life as in a major urban area, and is volunteers every week at the local library and gives tours, and takes public transportation, connects with her family all around the world via, was helping us with the Zoom connection that we were trying to set up with her. Just a remarkable person. And we talked about the benefits of people living longer, having Hilda's spirit, Hilda's productivity, volunteerism, her love for her family and for the community is a gift that I would love to bestow onto so many more people as a result of this research.
Terrific. Thank you very much. Zen, is there anything that you think we missed by telling this story?
Well, I think this is a really very important project, and it's a no-brainer that you should be doing it, and great for AFAR for sponsoring it. So we look forward to all the insights that will come from it. And I'm glad that you gave information about, for those who are interested in the project, they can Google on AFAR, AFAR, and it's the SuperAger project.
Initiative, SuperAger initiative.
Initiative, yeah. So
terrific. So now we move on, and then I'll need you here, and this is kind of the background. So we all know that that we have a chronological age and a biological age, and it's clearer as you get older, and I think people intuitively accept that. I think we also know that there's a huge progress in determining our biological age versus chronological age. The clocks were mentioned several times. It was about methylation clock, but proteomic was mentioned, and immunological clock and brain clock, and all those were mentioned. But what AFAR is very interested to do is not to tell you what's biological, to determine the biomarkers for biological age, but determine the biomarkers that will change when you practice geroprotection, okay, like exercise and diet, or gerotherapeutics, like some of the drugs like metformin, rapamycin, some of the other things that are being developed. And this is the challenge that probably will be validated by TAME, but we wanted to get a head start and really try to find a way to get those biomarkers sooner. And this initiative is also paid by the Schmidt Foundation and some others, and AFAR is leading that. I should say, and now you can ask me about that, but what I'd like to say is that I'm co-directing it with Jamie Justice, who I think was here before. I think Steve Krzyzewski have shown some of the data that she's part of the TAME study. She's a rising star, is getting the Rising Star for in AFAR, and is just wonderful to work with. And okay, and so what I kind of open it up. Tell me, what do I mean? Ask me what do you.
Well, very good. I'm glad to go for it, Nir. I think first of all, you are very creative in understanding that lab materials get collected during clinical trials, and that includes during trials of some potential candidates for actually modifying the aging process. So it sounds like you are going to take advantage of what's already in the freezer, so to speak, and maybe you could tell us a bit more how that would work.
So the first example is really simple. There was a study that was called the Diabetes Prevention Program, the DPP, where subjects that were not diabetics, but high risk for diabetes because of family history, obesity, um, were randomized to receive placebo, lifestyle change with exercise and diet, or metformin. The study was very successful, was stopped after 4 years because there were positive results, and the results were that both lifestyle intervention and metformin prevented diabetes.
Lifestyle had a larger effect size than metformin.
Right. Lifestyle lifestyle was 40%, diabetes was 30%. Of course, lifestyle was much more complicated and much more expensive because the people needed to come twice a week and exercise, and dietitians were involved. But lifestyle was terrific. The lifestyle also, they lost more weight than they did in metformin. So the idea here is that plasma is stored actually in two places. One of them is at the NIH, and some of it is within some of the principal investigators. But we would like to go and mainly see what happened in the first year of therapy. And the reason for that is that for the biotech, we really would like biomarkers that will change soon, right? Maybe 2 months in a phase 2 trial, and not do a phase 3 trial and wait 5 years for the results. So we're going to have both geroprotection, right, exercise and diet, and metformin in one study that we hope to get a lot of blood samples. And what we're going to do, we're going to do all the omics that you ever had in mind. We even are not committed to them because, as somebody said during the day, this is all already in progress. We just finished a 5,000 protein on 1,000 people, but less than 8,000 is not, they don't do it anymore even. So this is in evolution.
Just one subtlety, and maybe you could help us, and how you describe caloric restriction as a geroprotective mechanism. And certainly that does work in mice to increase lifespan, but caloric restriction can certainly improve more immediate metabolic parameters in people who have some metabolic disturbance that is leading them towards type 2 diabetes. So how do you ascribe the geroprotection to caloric restriction when it could also just be improving metabolic indices?
Right. So we're looking beyond the acute metabolic changes, right? We're looking for markers like methylation that are actually changes, not so, I'm quoting Steve Horvath and Morgan Levine, they're not changing so immediately. In fact, I don't think 2 months or even a year is a lot for them, but for proteomic it is. So let me just give you an example. It's a good question. How do you distinguish the metabolic from others? And they're totally linked. But let me give you an example. When we do our proteomics, we ask what's changing between age 65 and 95 in 1,000 people, and we get lots of things that are changing. But among them are a lot of breakdown of tissues, a breakdown of collagen, of matrix, degranulation of white blood cells, platelets are breaking up. There's a lot of breakdown. And initially, by the way, when I saw that, I said, that noise, I don't understand anything with this noise, but just to realize that maybe no matter what drug we're going to give, it has to stop the breakdown. So maybe you can get improving metabolism, okay, but not change in the breakdown. Mm-hmm. That's why you have to have omics that will really reflect something beyond just, because with, to improve your glucose, you can fast instead of 8 hours, 10 hours. You'll also get the results, right? So yes, we're talking about biomarkers that'll reflect much more of the hallmarks of aging than just the metabolomic changes that are more common. That's a very good point. The second, by the way, the second thing we're looking at is there's a new anti-diabetic drug, not so new anymore, since 2015, I think. It's called, the class is called SGLT2, and it's targeting diabetes. And then people notice that, yeah, you give it for diabetes, but it does other things. It prevents from cardiovascular disease. It prevents kidney failure. It actually decreases overall mortality, okay? And then when you give it to animals in the intervention testing program, they live longer and healthier. So this is a very strong, once you have a drug that targets more than one other disease and also change overall mortality, you kind of have the idea that this is a good drug. Now, in the last year, there are at least 5 papers in New England Journal of Medicine of what it's doing. And by the way, to diabetics and non-diabetics, okay? Right. So we're going to the PIs of those studies, and we're going to try and get, it's not in the pharmaceuticals don't have the plasma. It's the PIs of the study that have the plasma, and we're going to offer them a lucrative deal. We'll do everything for you, and you'll do whatever you want with it, and we'll do whatever you want with it, but we hope to get a lot of biomarkers that are going to be significant.
It's a brilliant idea. And as you say, it's taking advantage of what's available now, as opposed to doing long prospective trials that could take decades to really get the data in a form that would be useful.
So what we have a SAB that's composed of people who are either experts in every one of those metabolism or some of the leaders in the field. And what we need to find is, what do we have from those studies? But more important, which of the technology to use? Every time we're talking about it, there's a new technology. So we'll have to come up with a technology. We have to come up with the numbers of samples we need, with the price, because it's not going to be unlimited either. But we hope very rapidly, maybe, to make a lot of progress in the next year and started getting really down on the biomarkers. Now, I want to ask you something. I'm guessing that we'll have lots of biomarkers, and maybe we can need just few. Maybe at the end we'll need 100 or 10. I don't know. But what does it take? The fact that we have those biomarkers and we believe in them doesn't make it biomarkers for the FDA, right? I remember that the hemoglobin A1C wasn't a biomarker until every lab measured it the same way. So how do we eventually get to the FDA with biomarkers?
Right. Well, as you know, Nir, there is a process at FDA for qualifying biomarkers for different purposes, and not necessarily for predicting efficacy or the effect you're looking for, but also for selecting populations or for monitoring a particular safety outcome. So there are multiple uses of biomarkers. And it's true that what we're really looking for are biomarkers that will predict the desired response. And likewise, that kind of candidate would go through the qualification program. And the aim is not just to be a biomarker. Biomarkers in themselves aren't used for approving products. It's when they reach the status of a surrogate endpoint. A surrogate endpoint is one that is likely or does actually predict a clinical result. And so, as you mentioned, hemoglobin A1C is a great example.
Cholesterol, LDL cholesterol.
LDL cholesterol. And LDL cholesterol is interesting because it's true we approved the first statin, the basis of LDL cholesterol. That was actually my first new drug application review when I was just a newcomer to FDA back in the 80s. And it was on circumstantial evidence by and large. We had a few outcome studies where nicotinamide showed a benefit on CV results, but there wasn't really any convincing data that LDL cholesterol was the biomarker. We accepted it. We turned out to be right. Ultimately, the statins gave us a means of proving that lowering LDL cholesterol with statins actually produced the desired result, reduced cardiovascular risk. On the other hand, other agents other markers like HDL cholesterol, which we thought it was good to have high levels, turned out that drugs that actually raised HDL cholesterol typically, and there was at least one exception, they typically either showed no benefit or in at least one case actually caused harm through off-target effects. So HDL cholesterol didn't pan out, and that's the concern about not having definitive proof that clinical outcomes are improved by the therapy. So this has become more and more a formal process for FDA. That is, they want more evidence than we had when we made LDL cholesterol the endpoint, and we likewise when we chose hemoglobin A1C to reflect the benefit of lowering blood glucose. It's based on a lot of evidence, but not the kind of evidence that FDA today might want to require.
Zan, didn't Dave Fox yesterday say something about 1 out of 15 surrogate markers that the oncology division was looking at turned out to be validated? That's the kind of stuff that would give pause to the regulators, I would assume.
Well, that's right. Of course, that's in a different therapeutic area, but it has been a cautionary observation that the biomarkers that seem very plausible for predicting a particular clinical outcome don't always and frequently don't turn out to actually predict the benefit. And so that's why FDA has become a bit more hard-nosed about having evidence that a biomarker is actually something that could be used as a surrogate for approving an indication. And by the way, what kind of indication are we talking about? Well, I think we agree that there ought to be an indication for increasing health span. That I don't know how it has to be expressed on the drug package insert, but that certainly would be a very reasonable goal if, for example, the TAME trial shows that you can reduce the risk of multiple chronic diseases, and each disease is actually reduced in that composite. Well, then FDA ought to be willing to give some kind of indication. And we heard yesterday from some of our panelists that's a fairly plausible scenario, that FDA, if given those kind of data, they could approve such an indication. But that's what we're obviously striving for, a surrogate or multiple surrogates that could be used to approve products that will, we think, ultimately show an increase in health span.
So I'm even thinking more than that, because what we want to do is to prevent aging in time, not to get diseases, right? And so in a certain way, the biomarkers that I want are the ones that will be the indication for intervention and also the outcome, right? Yeah. That would be, but I'm not sure. So let me just, there's some on the chat, some things. And I want to first, Jamie Justice, who She told me she'll have to pick her son, but she was inclined to hear some of it. But she wanted to clarify that fast biomarkers analyses are not intended to replace prospective trials, okay?
Well,
But to
yeah.
provide a platform we can leverage while these trials like TAME are in process. And it's
Yeah.
absolutely true. I didn't mean...
That's right, Jamie.
I didn't mean not to. So I'm reading, I don't know what I'm reading, so I'll start and see. Can you better define healthy? A super aging with well-controlled diabetes? Okay, so what's healthy for super agers? So this is actually, Sofiya, are you still here? Can you answer this? Kind of what are the exclusion criteria?
All right. So I think John mentioned it, that for this particular SuperAgers study, we're really trying to cast a wide net because we are after 10,000 individuals. So the only exclusion that we will apply to this cohort will be people who have significant cognitive impairment where they cannot give consent to participate in the study, right? Because they don't have sufficient understanding of what is being requested of them. Otherwise, we don't have exclusions, but when we do data analysis, we'll be able to parse out, for the more healthy superagers who are the less healthy, because we're collecting very detailed medical history.
In our centenarian study, it was you had to be healthy, living independently when you're 95 years old. So it means that you had the cognition at least, and you were healthy enough. And then, yeah, some of them have diseases too, but that didn't kill them. Another question is, can people in other countries also sign up? And the answer is not yet. Sofiya doesn't sleep enough anyhow, and we don't need the world to contact her every time, but certainly it will be a thing. Now, Bruce Frankel, Bruce, you're there. Do you want to ask something?
Bruce was offering to help. Sorry, go ahead, Bruce.
Yeah, I wasn't so much asking. I was just saying that I'm happy to, and I contacted John already to say I'm happy to give a hand through the organizations that I participate in. And when I wrote my book, What Should I Do with the Rest of My Life, 13 years ago, we didn't call them superagers then, though we had them. So
right.
They were just people who lived a long time and continued to be productive. So
see, that's a retrospective analysis. It's good. It's good.
Near Marx Cantillon was asking, will families see results?
Sofiya?
Right. So we will be offering ancestry results. So we will be providing back genetic results for the individual's ancestry if they so choose, right? It will be an option for them. So that will be similar to what genetic services like 23andMe, right, and Ancestry.com provides a very detailed ancestry results. We're not going to be providing other genetic results because at this point, we're not sure what they mean, right? With time, as we conduct the studies, more research is going to be done and more information is discovered, then we will know better. But for now, we do plan to release ancestry genetic results.
What does it mean if we write to a centenarian, you have a longevity gene? What didn't he know? And what does it mean if he's 100 years old and we didn't find a longevity gene? So, but that was always our problem, that they didn't allow us to share results with the centenarians, because when you think of genetic results, you think of bad things, right? You think of BRCA, you think of some mutation, and we're looking for good genes, but the rules are the same rules, so we're not providing. Tom, anything else that you see?
Well, Candida is asking, are you collecting diet data?
We're not collecting particularly detailed diet data. We're just asking very basic questions. And the reason being, right, so we're recruiting people 95 and older. And I don't always remember what I ate for breakfast yesterday, So I'm not sure that they can tell me, right, what they've been eating over their lifetime. And diets change, and what they're eating now may not be what they were eating 30, 40 years ago. So we were not sure how reliable that information would actually be.
I remember
about-
when I was asked, when people told me when I started the Centenarian Project, people said, you should ask about sleep. You should ask if they're napping. And I went to the first guy and said, are you napping? He said, yeah, I'm napping. So I said, wow, that's amazing. So you're napping every day? Yeah, I'm napping every day. And last year I wasn't napping. And then you have to go 100 years to find, and they don't remember and they don't know. And diet is notoriously difficult to assess anyhow. So,
Any
and also,
assessment
I
of
think
physical activity, Nir? Are you asking patients about, or people, sorry, about their physical activity level or their sort of mobility, any way of characterizing how active they are physically?
Yeah, so that is of interest to us. So we do ask them questions about their current physical activity, their physical activity at the age of 50, just to kind of get a sense of how active they were, during their adulthood, and also asking very specific questions about their function currently.
We are on a balance between how much can we ask before they get tired of us?
Sure. Yeah, I
and
understand.
also the prospect that we would like to go back to them, because we would maybe like to get more than what just the swab we're getting in the future.
Yeah.
Right, that's a good point. They're actually going to become part of this community, and those who may be interested in participating in other studies in the future, there will be a registry for these people. So then if there are other investigators who want to do additional studies, then they can potentially reach out through this registry to these individuals and recruit them, enroll them in additional studies.
So you got examples of several projects and also the difficulties in doing them, but I'm sure that AFAR is going to help the field to move more rapidly with those initiatives. And, I would just say, look, some of those drugs, right, metformin, rapamycin, some of them, if you do it with exercise, maybe it'll push you a decade later, but human lifespan as a species 115 years, and we die now in the United States before the age of 76. It's declining. So there are 35 years that we can discover in addition. So I think these centenarian studies will give us more options, more drugs to target aging, and so we are very excited about that.
Well, it's great stuff you're doing, and AFAR, of course, is so important to the field. We've just heard about 2 of the programs, but there are many more. You support investigators and otherwise do a lot to accelerate the process of developing knowledge and products that can help us to live longer, healthier. So keep going. It's great stuff.
Thank you. We're targeting healthy longevity, just like the sign behind you says.
Well, we will use that term often, and thank you for all your support in pursuing that objective, Nir.
Looking forward to tomorrow.
And to Sofiya and John, thank you so much for joining us. It's great to have you, and we really appreciate it.